As you may already know, synthetic biology is the creation of new biological systems such as enzymes, genetic circuits or cells which aren't normally found in the natural world. The applications of synthetic biology include diagnostics, the environment, manufacturing and food, but in this video we'll be taking a look at regenerative medicine. The textbook definition is the process of replacing, engineering or regenerating human cells, tissues and organs in order to restore or establish normal function. The main categories are tissue engineering and biomaterials, gene therapy, and the regulation of stem cells. Let's start with tissue engineering. We have all seen news headlines such as "World's first lab-grown windpipe" or "Tiny lab-grown brains could cure Alzheimer's." But the truth of the matter is that organ growth is still in its infancy and remains both experimental and incredibly costly. However, one example of tissue engineering which is in use today is the fabrication of artificial skin. Traditionally, large losses of skin, say from excessive burns, were treated using autografts, which are skin grafts taken from the patient's own body. Grafts can also be taken from a donor or cadaver, although these methods carry a higher risk of rejection. In 1993, a US patent was granted for a form of artificial skin grown using materials like collagen. The process was granted FDA approval, but another group of researchers found that the resulting tissue lacked strength. To overcome this, the team used silk proteins harvested from golden orb web spiders. Spider silk is reportedly up to 10 times as strong as steel compared weight for weight and can stretch 20 times its normal size. Scientists used the silk to weave a meshwork alongside human skin cells in a bid to create artificial skin alternatives. It was a success, but using the biopolymer to grow skin in mass quantities was impractical due to the extremely difficult process of harvesting the silk. Besides being rare and difficult to work with, golden orb spiders are also cannibalistic and eat each other if kept in close proximity. As an alternative manufacturing method, Nexia Biotechnologies developed BioSteel, a fibre-based material made of the recombinant spider silk protein. What did they use to make the silk? Transgenic goats. They harvest the fibres from the goat's milk and then use chromatography to purify the silk proteins. Other applications of artificial silk biopolymers include implant surface coatings and the construction of artificial ligaments or tendons due to its elastic tendencies and biocompatibility. However, no one has yet been able to produce enough of the silk in large commercial quantities. Utah State University are still researching and developing the technology and currently have 30 spider goats at their university-run farm. Another treatment which is currently on the market for scars, burns and hyperpigmentation uses the patient's own cells in a regenerating process which accelerates healing, minimises scarring and reintroduces pigment to the skin. A small biopsy is taken from an area close to the skin being treated to ensure that the texture and colour are the same. The sample cells are then separated to produce a spray solution which contains all of the cells necessary to promote healthy skin growth. This includes keratinocytes, which are the cells that make up the outermost layer of the skin, and melanocytes, which are the melanin-producing cells. These are then sprayed onto the affected area where they multiply and spread throughout the skin, resulting in increased healing and restoration of skin colour. A well-known cosmetics company currently grows skin samples from tissues donated by plastic surgery patients. It produces more than 100,000 half a centimetre square samples per year and grows 9 varieties across all ages and ethnicities. The use of animals for all sorts of product testing and research has long been a contentious issue. However, as advances are made in the field of synthetic biology, this matter could be very close to being resolved. Now let's look at an example which uses genetic engineering and gene therapy. Cystic fibrosis is an inherited life-threatening disorder that damages the lungs, digestive and reproductive systems. It is most commonly caused by a mutation in the cystic fibrosis gene. The gene affected controls the movement of salt and water in and out of cells, causing a buildup of thick, sticky mucus, which dramatically increases the risk of infection from bacteria such as Pseudomonas aeruginosa. The disorder has no cure, and therapy typically consists of symptom management rather than treatment. As cystic fibrosis is a genetic disease, can we use genetic engineering and synthetic biology to treat it? Obviously, we can't heat shock or electroporate humans in order to make them take up foreign DNA, so how do we get around this? Gene therapy is being developed as a novel treatment for cystic fibrosis. This involves the transfer of correct copies of the cystic fibrosis transmembrane conductance regulator DNA to the epithelial cells in the airways. However, as cystic fibrosis is a lifelong disease, turnover of the airway epithelium means that repeat administration may be needed. To overcome this, lentiviral vectors are being used to transport the functioning version of the gene into the human host. This is because lentiviruses actually integrate into the host genome and allow long-lasting gene expression. The first in-man lentivirus trial in patients with cystic fibrosis is currently underway, and if the trial is a success, the same technology could be used to treat other genetic diseases such as sickle cell anemia, haemophilia, and spinal muscular atrophy. Lastly, here is an example of how stem cells are being used in tissue regeneration. This friendly little guy is an axolotl, which is a unique type of salamander found only in Mexico. Many amphibians have the ability to regenerate limbs, but axolotls blow their competition out of the water with their intense ability to regrow not just limbs, but their tails, jaws, skin, and even spinal cords. They achieve this with zero scar tissue, and it doesn't matter how many times you chop off one of their arms, it will grow back perfectly every time. This is thanks to pluripotent stem cells migrating to the site of injury. These cells have the ability to differentiate into any type of cell. Embryonic stem cells are considered pluripotent, but we lose these as we age. This is why if a mammalian fetus loses a limb within the first trimester of pregnancy, it will regrow that limb. Induced pluripotent stem cells are a type of stem cell that can be generated directly from adult cells. In 2006, a lab in Japan found that the introduction of 4 specific genes encoding transcription factors could convert adult cells into pluripotent stem cells, potentially negating both the practical and ethical issues which surround the use of embryonic stem cells. As they can propagate indefinitely, as well as give rise to every other type of cell in the body, such as neurons, heart, pancreatic and liver cells, they hold great promise in the field of regenerative medicine. Scientists are currently working on how to deliver them to the target tissues accurately and in a non-invasive manner. Interestingly, liposuction fluid is a great source of stem cells. Patients could potentially be treated with their own adipose-derived stem cells, or the cells could be used in other tissue engineering applications. Perhaps carrying a little extra weight isn't so bad after all. These are just a few examples of the infinite applications of synthetic biology to regenerative medicine. We hope that you found it interesting, and please let us know if you have any questions or queries. Thank you very much for watching. This video is only possible due to the generous support of our sponsors, BBSRC, the Wellcome Trust and the SEB, which we'd like to thank at this point. If you'd like to find out more about our project, check out the additional videos on this channel.